We're thrilled to welcome physician scientist Mark Chao, co-founder and CEO of TenSixteen Bio, to the show today. Thank you once again, Mark, for joining us.
Thanks, Chris. I appreciate you guys for having me.
Absolutely. Let's kick things off. Mark, can you share a brief personal intro with us?
Yeah, absolutely. So my background, I'm a physician scientist and actually a hematologist by training. So grew up in the East Coast, but slowly migrated my way out to the West Coast. Did college in Ohio, but then landed at Stanford, where I spent about probably 15 plus years. So did my MD there, PhD in cancer biology in the lab of Irv Weissman, who's a big stem cell biologist. And then did all of my clinical training, including hematology fellowship. Interesting enough, thought I had my academic 40-year plan, thought I was going to be, running a lab and seeing patients, and had planned this out, like most medical students, 15 years ago, but really got into actually at that point a transition into industry. And so I actually co-founded a company, my first company called 47 Inc., which is an immuno-oncology company that worked on a first-in-class therapeutic targeting macrophages. And it was interesting, really tough decision for me to leave academia to go into industry, but a lot of the drivers for me really were centered around how I thought I could make the most impact to patients, and really thought, we were discovering a lot of things in the lab, but drug development really happens in industry and companies. So really wanted to jump into that. So co-founded 47, headed up clinical development, was a great ride. So we ended up going public just a few years after we founded, and then grew to about 80 employees. And then were acquired by Gilead Sciences for about a little less than $5 billion 2 years ago. Spent about a year plus in the Gilead oncology leadership team. Really loved the experience building out the oncology portfolio, getting a sense of how large pharma works. But always knew I was going to go back to biotech, really love innovation, really love building teams. And so had synced up with Foresight Labs, which is an incubator arm of Foresight Capital, around TenSixteen and the concept, and so joined as one of the co-founders and CEO of TenSixteen Bio. So that's a little bit about me. I would just say the high-level story is, if you asked me 10 years ago where I'd be, I couldn't tell you this is where it was, but that's kind of the fun of careers in a biotech, as you guys know.
Absolutely. And you've actually prompted me to ask something a little bit off script, but I'm curious, why 47? Why TenSixteen? Why the numeric names for the startups?
Yeah, it's a great question. Some people now sense a pattern in terms of naming things after numbers. So for 47, that was named actually after the target of our lead asset, which was CD47. I'll say, and it's a good segue into TenSixteen. TenSixteen actually is named after the number 10 to the 16th power. So 1 with 16 zeros. And that highlights a little bit about the science we'll get into, but it's actually the estimated number of times a cell divides in a human body's lifetime. So you think about it, it's a number that's astronomical, and gets at a concept around central to TenSixteen, around the fact of genetic change, and how each cell division represents an opportunity for genetic change, for mutations, and that creates different disease risks, again, which we'll delve into, I'm sure, in more detail.
Absolutely, and that, as you said, segues perfectly. Can you give us, and you've started to, just a brief overview of TenSixteen before we start to explore a little bit further?
Yeah, absolutely. So super excited about this company. As I mentioned, was an effort out of Foresight Labs, which is really an incubator arm that focuses a lot on the merge of technology and healthcare science, big data, really innovative concepts. And so TenSixteen, at our core, we're a precision therapeutics platform company targeting this broad biology of what we call somatic mosaicism, or changes in your genetic code in different populations over time. And the first application of that, or somatic mosaicism, actually occurs in the blood through a process called clonal hematopoiesis, or CHIP. This is a broad area of biology about less than a decade old, which we'll talk in more detail, but is really foundational to modifying disease risk in multiple disease areas. So we're looking to target this broad biology, therapeutically develop drugs against this, and to really change the treatment paradigm, as we'll talk about more. We launched, I think this concept has been something that has been worked on over the last couple of years, but formally, I think, launched out of stealth and made a public announcement just this past February, so a couple months ago.
And congratulations on that. And thank you for being willing to join us and speak about it today. Michael, over to you. Let's dive a little bit deeper.
Yeah, thank you, Chris. And thank you, Mark, for the intro as well. I'm excited to learn a lot more. In February, as you mentioned, TenSixteen launched out of Foresight Labs, and you had a really bold vision for treating age-related diseases with precision medicine. So I think you had set the stage a bit well for it previously. Can you now share more about the origin story of TenSixteen and really what was that spark that led you to founding it?
Yeah, absolutely. So it's as I mentioned, you'll see some themes here where I think probably two things. I think one is a theme around innovation, and then one around serendipity. So for me, kind of my personal involvement into TenSixteen probably spanned, even about 8 or 9 years ago in terms of the concept. So this concept of clonal hematopoiesis, which again I'll talk more about, was an area of biology that was founded about 8 years ago. As a hematologist and as a stem cell biologist, it was an area of biology I was incredibly interested in. And briefly, what we know, even from those seminal, discussions or discoveries just even about 9 years ago, is that as we age, we develop mutations in our blood stem cells in cancer-associated genes. These are called CHIP genes. So there's about 10 or 15. And we now know that when you have a mutation in this gene or these genes, you develop a significantly high risk of multiple diseases. So cancer, cardiovascular disease, autoimmune disease, and the list goes on. And it was really over this last 8 or 9 years where this biology started to be really central, really broad, and really causal. But yet today, nobody had really solely focused on therapeutically targeting this. And so that was, as I was following the biology, was super intrigued, and then linked up with Foresight Labs through some mutual connections about a couple of years, or actually about 1.5 years ago, where they were also looking at this idea. Kind of, again, their theme is how can we think about the next areas in healthcare and science and really develop something that's groundbreaking. And so a group at Foresight Labs, again, led by Vic Bajaj, Jim Tannenbaum, and others, were exploring this idea similarly. And so we had caught up, and I was surprised to see that, a group was already thinking about this pretty broadly in terms of, how to be kind of a first mover in this space of exciting biology. How do you think about, developing drugs and thinking about the end-to-end ecosystem? And so it was about, 1.5 years ago, some informal discussions that led to more formal discussions as we were building out the concept and saying, okay, if we're going to be one of the first, if not first in the space, when is the right time? Are we too early? Are we just right? How do we think about building differentiators for success? And so about, maybe over 9 months ago, kind of joined in a more serious way, and then ended up coming in and starting as CEO and co-founder. And then, the rest is history. So it's been really exciting, been working full time for several months now, building the team, building the vision. But again, it's been a really incredible experience so far.
Yeah, that is an exciting story. And it's incredible to think as a first mover, you're following 8 to 9 years of science, like really innovative science. So just taking a moment to, recognize we do stand on the shoulders of giants in this industry, which it's an incredible privilege to be able to do that for human health. But to take another step forward, after you had joined with Foresight Labs, can you maybe explain a little bit about kind of what that experience was like and what value that Foresight was able to have for TenSixteen in its earliest stages?
Yeah, no, absolutely. I think it's a great question, Michael. So I'll just say again, for me, as you can kind of get a theme, I really like working on things that are really transformational. So either first-in-class drug products, or in this case, really a first-in-class treatment paradigm shift. And that really, again, is what TenSixteen, fits in a lot of ways. I would say that during my time at Gilead, or even before that, I was kind of thinking about, what were my next steps, and talking to various folks, in terms of, new companies and so forth. And I think one of the things that struck me specifically about Foresight Labs was really their expertise and focus on large-scale data, large-scale genomics, and kind of this idea of how do you really take these different sources of data that now we have access to, sequencing data, clinical data, biomarker data, and synthesize it in a way that's usable in innovative ways. And that's really, I think, one of Foresight's really core expertise in terms of the people that were brought. Is an area of expertise that actually is complementary to kind of my own, which is, clinical development, hematopoiesis biology, translational research. So that really drew me to that. And then I think really similar, we thought in very similar ways in terms of big, bold ideas, but bold ideas that can be accomplished really in a time frame that's really feasible. And so again, going back to this concept of 1016, that all those ingredients, where you could work on a field in an area of biology that we both believe is going to be incredibly ripe for transformation. But do it in a way where you're first moving, where you develop a large advantage, where you integrate data into kind of clinical development, developing therapeutic targets, and think about the whole healthcare ecosystem in a way that a small biotech can actually accomplish. And so those were all the ingredients that I think were really made for a great relationship. I've really enjoyed working in that type of setting. And again, I think stepping back a bit more, for folks thinking about working with different incubators, a lot of it is just about kind of what expertise and synergy can you guys bring together? And this was one of those cases.
Yeah, well, it makes complete sense. And especially on this topic of somatic mosaicism, having a very kind of sophisticated data first approach would be extremely valuable. So at this point, let's dive a little bit more into the science. I'm really excited to learn more about somatic mosaicism, this new area of science, and how is it that this new understanding is leading the way to new avenues of medicine? What is 1016 doing?
Yeah, no, thanks. So, I'll start off pretty broadly. Again, as I mentioned, 1016, we're a precision therapeutics platform company focused on somatic mosaicism first. And I want to spend a couple of minutes there because I think this is kind of illustrates, as you mentioned, Michael, kind of why we're excited about this. How does genetics play a role? So we know, obviously, that, genetic drivers are really important in disease. Traditionally, our genes are inherited at birth, right? So we have a lot of, germline genetically predisposed conditions, that folks are working on. However, we know that a large majority of diseases, including those that are associated with age, actually occur in genetic changes after birth. So what are called somatic mutations, so mutations in our somatic cells. This idea where you can have genetic mutations in different populations of cells essentially is the concept of somatic mosaicism. And again, as I mentioned, when you have these mutations in different cell subsets, this can lead to changes in your disease risk. So every day as you age, your disease risk changes because your personal genome is quite dynamic. And I think it's an area that is starting to be tapped. I would say as an oncologist, this happens frequently. So in the oncology world, we're quite familiar with this concept. But outside of oncology, I think there's a growing appreciation that these changes in genes over time actually modulate significant risk, whether it's heart disease or autoimmune disease or liver disease or others. And so you can think about these changes in different cells, whether it's in the blood with CHIP or in other tissues. Therapeutically targeting this change is relatively in its nascent phases. So this is how we think about the broad opportunity where, again, there's this whole biology that exists as we age that we really need to target and incorporate when we think about who are patients at highest risk for treatment. How do we develop drugs? So that's, I think, a broad view. We're very interested in this broad view of somatic mosaicism. And then how do we think about kind of the first point of entry, right? How do we focus as a biotech to provide, near-term value? That's where we focused on the blood, because probably the best example of somatic mosaicism that's most validated is in the blood through this process called CHIP that I mentioned. So our initial focus is on CHIP, or clonal hematopoiesis. And again, as I mentioned, you have mutations in your blood cells, and then they create significant risk of disease, to the point where if you have these CHIP mutations and other factors, you can have as high as a 50-fold increased risk of disease, of developing disease, like cancer or heart disease. And so I want to pause there just to interject kind of what is 1016's vision? Why are we doing this? You can kind of appreciate that there's this area of broad biology, but for all of us in the company, it's about how do you bring that to the patient, to the individual. So I shared one story during our launch that again was really impactful for me. So as a hematologist, my specialty before I went into industry was in aggressive blood cancers. While I was training at Stanford, I remember early on in my training, having cared with many folks with a disease called acute myeloid leukemia, or aggressive blood cancer of the white cells. It's a devastating disease. It is a disease where less than half of the patients, even with our treatments, get cured, where we're still using the same drugs we used 40 years ago. So a lot of really high unmet needs. A lot of times when we saw patients at Stanford, they already were referred to us after had multiple prior lines of therapy. And at that point, you can imagine, if you have so many different therapies, the disease has already developed mutations and resistance. So there were 20 or 30 mutations that we saw. There was one person that was referred to us who initially had treatment, got into remission with a bone marrow transplant, which is really the only effective cure. But then about less than a year later, relapsed with aggressive disease, went from 1 mutation to 10 mutations. We treated with a couple more therapies that really didn't work. And we were stuck in a scenario where we had nothing else to offer. And I remember that was a specific point where that amount of frustration and hopelessness as a physician is one of the worst feelings you can have in terms of being able to say, what else do we have for you? That patient ended up passing away just a couple months later after that conversation. And unfortunately, you can think about several patients similarly. And so I say this because I bring it back to a concept where, what if instead of treating patients when they've had 10, 15 mutations, when we're treating 6-line therapy, how can we move up in the treatment change to treat disease earlier where there's less genetic complexity, where instead of 10 mutations, there's 1 mutation? Is there kind of a precursor state that we can treat to, in some ways, ideally prevent the disease from even occurring, or at its best case, at least significantly prolong that? And so that's really what our mission at TenSixteen is to treat disease at its earlier stage. And we do that through CHIP, for example, as a biomarker, as a precursor state. And you can see ramifications of this. I think when we think about treatment paradigms, this is a huge treatment paradigm shift to be able to do this, not only across cancer, but across multiple diseases. And it's a treatment shift I think is very real and very immediate if you think about drug development in the right way. So that's the type of thing I think we're involved in. We see it as really bold, but again, an actionable path. And that's what gets us, including myself, excited every day at TenSixteen. So that's a little bit about the why, I would say. And I know, Michael, you had mentioned, okay, so let's talk a little bit about the how. How are you doing this? And this is where, again, it comes in with kind of this focus on data. So if you think about how do we target or develop therapies for CHIP, you really have to think about an ecosystem. It starts with, number one, how do you identify the therapeutic targets? So we're developing what we'll hope to be one of the most, if not most extensive, clinical/multi-omics databases for CHIP. And so that includes sequencing, that includes methylation and other transcriptomics, coupled with clinical data to be able to do 3 things. I think one is to identify who are those patients at highest risk that have CHIP that need intervention. Number 2, what are the best therapeutic targets that we can leverage for CHIP biology? And then number 3, how do we develop a biomarker so that when we identify these patients, we can do it in a precision way for regulatory approval? So for our platform, it's very prospective. We're aggregating, obviously, public data, but more importantly, leveraging collaborations with academic centers, healthcare networks, to really create a significantly large scale of data that we can pull specifically for CHIP. So that's kind of our engine driver. And then as we identify targets, we develop therapeutics. And then again, we pair that with a high-risk population with a biomarker, and then look for, obviously, a track to clinical approval. But then also thinking about how do we think about how physicians and patients are going to use this for early detection and so forth. So that's where we see the whole ecosystem. And then the last thing I'll say before I pause is it all starts with people. I feel like this is a great, bold idea, but you equally need not only great science, but great people. And so again, I think this is where, again, I'm personally very excited. It starts with the co-founders that we're working with, who are really an incredible group. There are 4 co-founders and then 2 founding SAB members that have really led the field of CHIP, super passionate. It's been really a joy. And then the team that we've built in terms of diverse skill sets to really accomplish this goal. So that gives you a little bit more under the hood about what we're doing, and certainly can go into further detail too.
Actually leads well into a question I had as you were speaking, Mark. How does the technology you're talking about, chip, somatic mosaicism, fit into the landscape around hematopoietic stem cell replacement therapy and gene editing that's becoming more prevalent today?
Yeah, Chris, it's a really interesting question because I think there are applications to different fields, as you mentioned, gene therapy, regenerative medicine. And so going back to the biology, as I alluded to, so chip mutations, they occur in our hematopoietic or blood-forming stem cells. And the reason why that's important is because hematopoietic stem cells are the only type of cells in the blood that can produce this exact copy of the cell and delineate all the different cell populations of the blood. It's the only unique cell that can do that. So it has a lot of properties. When you get a mutation in that hematopoietic stem cell, it can have drastic consequences. That's what we see with chip, right? Where we see, multiple diseases at high risk because of this one mutation, this one type of cell. And so that's one angle in which we're looking at chip, but it applies actually to other areas. I'll say first on the clinical side, we know that especially in oncology, one of the cures that are used in blood cancers is a hematopoietic stem cell transplant, where you take, in a lot of ways, a healthy donor's blood stem cells and transplant them into a patient with disease, where you first wipe out the disease and then replace their bone marrow with that. That's, again, our only known cure for several heme malignancies. Now, why I mention that is because even when you do that, there is a risk for relapse. And a lot of those relapse are in genes or mutations that are chip genes, whether they come from the donor itself or the host. So there's this whole biology, even in stem cell transplants, where we can leverage and therapeutically target chip, number one. And then you mentioned about gene therapy. It's really interesting. I think when we think about certain modalities, and certainly we've thought about this peripherally, is there are possibilities of using gene therapy approaches for targets like chip, because it's so foundational in that way. So we've been thinking about our therapeutic approaches, primarily small molecules and antibodies, but there are other modalities that can be used, including gene therapy. So it really touches, I think, a broad area. That's why we really like the idea of chip and somatic mosaicism, is there's so much white space. There's so much, even what we say is low-hanging fruit to be able to target and then go into, broader ideas.
Thank you for sharing that with us. And I think let's take that step back then and chat a little bit more about the therapeutic platform. So you've given us a great insight into your data-driven approach into the biology. Can you tell us more about TenSixteen's drug discovery and development platform and share how it differs from traditional genomics approaches?
Yeah, absolutely. So again, as mentioned, we think about that end-to-end ecosystem. And so picking on that, I think there's a couple of things that really are important for what we're doing. Number one is an ability to identify targets specific to chip biology. Some of these targets are known, but a lot of these targets either are unknown first in class, or even targets that can be repositioned to a very high-risk population. So at kind of a high-level core, you can think about chip as really a broad universal biomarker. I can't think of many other types of even genetic mutations that lead to that type of broadness. So that's number one on target identification. And our data platform with specific proprietary analytical tools and other aspects and even preclinical models allows us to really differentiate there. In terms of the therapeutics development, I think that then feeds into our internal engine. Again, as I mentioned, primarily focused on small molecules and antibodies. For a couple of reasons. I think when you think about treating earlier, dosing convenience and so forth is actually really important. So that's one factor. But I think the other piece that's important is, other than just saying, okay, we have a target and we have a indication, let's say it's blood cancers. For us, that's not enough. We need to take it a couple of steps further to say, who is the exact patient population that has chip that is at highest risk? There are some people with chip that will have a very low risk of developing certain diseases, some that will have nearly 100% risk of developing, for example, cancer in a couple of years. So the latter example are the type of people that we're looking to identify. And again, that's fed through our database. So you not only have a target and a drug, but you pair that with a high-risk population. And because we're in a precision type development, we know for regulatory approval and for diagnostics, you need a companion diagnostic or an assay that's able to survey for those that are CHIP and that have these risks. So we're simultaneously developing that early on, again, to pair target with patient population with biomarker. So for any of our development programs, those three ingredients have to be in place. And I think that's where, again, we can take a very hopefully high probability of success approach, but a very defined approach to really get at those that are at super high risk.
And then that's exactly the sort of ecosystem play and platform that we also find so incredibly exciting in the tech bio space right now, taking that real world information, developing a more targeted patient population and associated companion diagnostics, and then figuring out, all right, now that we know who these patients are, how can we get them the best treatment possible? So absolutely right there with you. And talking about that platform, then taking it into the future, how do you think about, as our understanding of somatic mosaicism evolves, not only helping drive that change, but evolving TenSixteen with it? How do you think about innovating the future of somatic mosaicism?
Yeah. I mean, I think I'll step back and again, an area that gets me excited when you think about kind of new fields and trying to really move the needle. I think we're really privileged, to have our academic co-founders, but to be a part of the therapeutic innovation here. I think it's early enough where now people are starting to realize, okay, CHIP is real. It's pretty broad, but how do we think about targeting these? And I think having been part of an earlier genesis with 47 or CD47, when we started, we were targeting macrophages. People thought at that current time macrophages were bad. They only increase tumor risk. Why would you target it? Our target CD47 was expressed broadly in the body. So many people were like, you guys are idiots, like this isn't gonna work. And it was a lot of skepticism, I think, at the initial part. But now, over that last 10 to 15 years, there's now, I think, over 100, probably more companies working on CD47 as a target. The fields recognize that innate checkpoints are important. And I use that example because I think this is also an area where CHIP is now, where there's a lot of excitement, but then how do you drive towards really making it into therapeutic reality? Ultimately, that's the end of the day of what we're doing. And so I anticipate, as we're thinking this broad, there are going to be some skeptics. How do we think about overcoming challenges that we've thought about? But really the horizon is quite amazing in the sense that I think over the next several years, there will really be a growing appreciation that this is a huge area of biology. Now, specifically to your question, Chris, I think, where do we see kind of that evolution occurring? I mentioned again that our first application is in CHIP in the blood, but I think where you're going to see innovation is this broader concept of somatic mosaicism. So how can we find these genetic changes after birth in other tissue types? We know that this occurs in the GI system, in the ovarian system, in the skin. And those efforts of understanding those changes in therapeutic targets is relatively in its infancy. But I think as we're one part of the equation in that, our database is going to leverage to try to develop tissue-specific somatic mosaicism in other tissue types. I think we're going to be one of many folks that are going to be interested in this area of science. And so I would anticipate, I think what's great is, to be able to bring spotlight to this field so that others can come in, because there's so much need and so much innovation here. It's really going to take multiple companies, multiple ecosystems to work on that. And again, we feel privileged to be one of the first to go into the space.
I think we all benefit from it. So thank you for taking that leap and helping be the first movers. It's always an exciting, but in some ways challenging position to be in. So we all benefit from it. Thank you, Mark. And shifting slightly, let's talk a little bit more about age-related diseases. Can you tell us in this space, what is TenSixteen working to address, and why is now the time for exploring that treatment of age-related disease?
Yeah, I mean, I think it's been well known that there are many diseases associated with aging. Probably some of the best examples of recent coverage have been in neurodegenerative diseases like Alzheimer's and so forth. But we know that it actually is the majority of diseases. So cancer is primarily a disease of aging because cancer relies on genetic mutations. And the longer we live, the more divisions we have, the more mutations we can get. We know cardiovascular disease is also a disease of aging. We know certain autoimmune diseases are too. So I would say this concept of age-related diseases has been around for a while. I think what's different, or at least where we're trying to focus on, is there are multiple factors in how one gets a disease associated with aging, right? Environmental, genetics, other factors, epigenetics. And I think where we're trying to really focus is to say that we think that genetic drivers are one of the strongest drivers of disease. We know that from multiple examples. So in this case, for age-related diseases, how do we focus on that driver and separate that driver from some of the other environmental factors, which we know are important, but finding areas where we can really assess the genetic driver instead. So that's kind of the area of which we're looking at. I think for us as kind of initial focus, there are a lot of diseases that actually, for example, CHIP plays a role in. And for us as a biotech, we obviously have to focus, how do we prove the concept and then look to expand? And our initial areas, for a couple of reasons, I think are focusing, number one, on oncology, and then number two, in cardiovascular disease. And part of the reasons for both of those is because that's where a lot of the science has been best validated. That's where we see kind of the most opportunity to really target this biology and to think about clinical development paths that can be achieved, especially in oncology, for example. The unmet need on cardiovascular disease is how can we think about really stratifying a very high risk population through genetics. That's starting to get appreciated, but for the most part, we treat, cardiovascular disease patients or those that are at risk for heart attack very broadly. So there are ways to think about, super high risk patients in that case. And you can see that value and how we do that is going to be different from each indication. So that's kind of a sense about how we're looking at the initial pieces. I will say that as we're, developing data and internal insights, there are several other diseases we're quite interested in, and it's just about how much time and focus and capital do we have to really devote. But again, there's so much white space here. For us, it's really about taking a stepwise approach.
Well, when you're finally expanding, we'll have to have you back on the podcast so that way we can
That's
talk.
right. We'll talk about all the other indications we're hopefully going after.
For now, Michael, please.
Yeah, so Mark, you earlier had very briefly overviewed your personal background. And so we'd like to, take a step out of TenSixteen and shift back over to you. You very humbly skimmed over the fact that you had co-founded a company which, was very, very innovative in general, but especially for its time, was met with a lot of skepticism. And despite it being your first kind of company which you founded, your first foray into the entrepreneurial space, it had succeeded and sold for around 5 billion, which is incredible. And so kind of throughout that journey and throughout the journey so far at TenSixteen, which has, sure had its own unique challenges, what were some of the greatest challenges that you had to face as an individual transitioning out of perhaps more of an academic setting, and then just in general?
Yeah, Michael, I'm glad you asked that question. I can tell you many stories here, but I will say a couple of things. As you mentioned, I had my academic plan the whole way, and 47 was my first foray into industry. So I didn't know anything about industry and then was vaulted into this. And so you can imagine, a lot of mistakes that were made. I would say this, I think one of the things that was really insightful for me necessarily wasn't around learning drug development or industry, which obviously is a challenge, but was actually more on the people front. And I think really one of the things I learned was, how can one be a really great manager? How important are the people in terms of driving a vision? And so I'll give you a little context. When I came in to 47, I thought I was a pretty good manager. I had managed teams in the hospital, residents, medical students, lab folks, so forth. I would say generally pretty friendly, at least hopefully. But I really was very insightful when I came into industry, understanding that everything is so team-based. How do you think about all the different stakeholders and personalities to be really an effective manager? And it was something where I learned a lot of lessons. I think for the first part, I was kind of saying, well, I'm used to doing things a certain way, but now I see there are 15 ways to do it, different perspectives. How do you kind of balance those things? How do you make sure that for everybody you're working with, either as a peer or in a matrix environment or your direct reports, how do you motivate them and really capitalize on their strengths, but also think about career growth? So it was really, I think what I learned a lot of my time at 47 was very fortunate to have worked with a lot of folks, operators that were really experienced. On our executive team, we had folks that were in the industry for 20, 30 years, had built, an organization from 50 up to 2,000. So I learned a lot of insights of how do you scale culture. How important is positive culture? Not only when we were 20, 25 people at 47, where everybody knew each other's names, to 50, to 100. How do you, and then, for other companies beyond that, 500, 1,000, how do you scale that? So I learned a lot of, I think, intentionality about just great kind of behaviors and aspects really from people. And if there's a theme kind of in my story of how I feel like I've been lucky, is been having great mentors, both from the academic side and the industry side. I've been very fortunate. So that was probably the biggest eye-opening pieces in kind of the early days. And I say how that translates because when we think about 47, I'll give you some examples. Drug development's hard, right? So you're always going to face adversity, and 47 was no different. When we went public several years back, we're a clinical stage company. We were developing actually the drug in non-Hodgkin's lymphoma. IPO'd, and then we had some data readouts that looked different from our phase 1 to phase 2 data. And so that actually changed the perception on whether the drug worked as well as we had thought. Our stock price went down from $16 at the IPO to a nadir of like $5 or $6. And so there were some, hard discussions about how do we finance to continue that piece? How do we continue to highlight the potential of our lead asset? And long story short, I mean, really what it was that the people in the room, we were all aligned on a vision. We faced adversity, but we knew always that there was going to be a sense that we were going to figure it out and pivot. And so we pivoted to another indication. These were actually in a type of blood cancers, where we started to get really great data. And then we were able to highlight to Wall Street and to other stakeholders and investors how important these indications were, how there was an unmet need that was really huge, how our drug was addressing it. And from that time period where our stock was $5, it was about 9 months to a year where we then had critical data readouts. Our stock popped to $25, $30, and then several months later we're acquired, ultimately for $90+ a share. I use that example just to say that we had faced hardship like anybody else, but again, it's really about the people. And so those are lessons that I've taken, certainly for TenSixteen. One of the reasons why I decided to jump in and become a CEO was a couple of things. One of it was to really build great company vision and really build a culture where you foster the excitement, you can align everybody around one singular vision. And there's just something really special about that. I think at the end of the day, when you get a group of people tackling a really hard problem that are all marching in the same direction, it's amazing what can be accomplished. And ultimately, I think for all the challenges of being a CEO, that's the one thing that really kind of continues to drive me. And again, it's early at TenSixteen, but lessons that I've learned, at least in my short career.
Well, I appreciate you sharing about the kind of people aspect. I think it's really often overlooked when kind of considering the challenge of trying to lead teams through developing drugs, which is, as you mentioned, just such an incredibly difficult thing to do. So kind of playing off of that question then, knowing what you know now, having gone through what you have gone through and seeing the challenges that you will eventually have to overcome as a medical student, what advice would you give to yourself, or what do you wish that you would have done differently? Maybe not wished, but could you have gone back? Would you have done anything differently?
Yeah, it's an interesting question. The short answer is yes. I think there are definitely things that I learned around the way, but I'll answer that a little bit differently. Is that I'm really glad for a lot of the things I would say you could call them, certainly in my career, certain failures or challenges. And had that not happened, I wouldn't have learned a lot of things. In some ways, and this is a little bit cliche, but I've learned far more from the adversity and the challenges than the success. Absolutely. I mean, I think probably 95% of what I've learned have been in those things, because really when things are going well, you're not pushed to kind of anticipate the challenges and really kind of manage crises and think about really why you're doing things. So I'd say like a lot of the things that I might have done differently, I'm actually glad happened because it was really important for my development. But I would say for like my younger self or self back in training or for others, one of the things that I think I would prescribe advice is just to be open to opportunities and flexibility. As I mentioned, I mean, as a medical student, I was, okay, in year 4, I'm going to go graduate and then go into residency and then fellowship and then faculty and do this. you kind of have these blinders on of what you're going to do. And Stanford was a great place because it allowed flexibility and there's just so much going on. But I didn't realize, I think it took me a while to realize what else was out there besides the academic track until later. Really, it was actually the 47 experience. I think had I been a little bit more open, I would have seen those things out a lot earlier. So that's probably the one advice I would give is just, be open to opportunities. You never know where things go. Again, as I mentioned, if you asked me 15 years ago whether I'd go into industry, I would have said no. If you asked me, 5 years ago, would I be in the position I'm in, I probably would say no, I wouldn't think that. So it's one of the beauties, I think, of certainly for me, being at peace with that type of change and opportunity. And I think it just allows, just so many really cool things to happen. I think that you never thought you would be interested in as long as you're open to it. So that's probably the biggest advice, is to be open. Every, I think, story of an entrepreneur or somebody who's done things, there's always luck and serendipity. My story is no different. And I think, part of it is you create some of that, but part of it is also just, being open to when those chances go and taking them.
Yeah, I think that's great advice. And I could even say to myself as well, you have to be open to the opportunities, because it's luck in hindsight, but in prospect, it's the opportunity which makes the most sense, and you have to stay kind of open-minded. So, appreciate you kind of sharing that. Thank you again for sharing about kind of the past and near future of TenSixteen. And so we're now hoping to turn the dial forward more into the future and kind of return to the discussion of somatic mosaicism and its role perhaps in kind of the future of medicine. So you mentioned that it really is bringing forth a new paradigm of medicine. And so we would love to explore, what does this new paradigm look like? Sure, we're moving towards preventative treatment, but could you maybe flesh out that vision a bit more for us?
Yeah, I think kind of if I think really broadly, kind of in that type of horizon, it really is simply, how do we detect, intercept, and treat disease at its earliest stage? I think that's the broad vision. Somatic mosaicism and CHIP is one route to do that, but I think there are other efforts to think about that. We know, for example, that many companies, including large pharma, have moved to earlier stages of treatment because that's really where you can create the most foundational change. And there are multiple methods to do that. So to me, it's really that aspect, is early detection, early treatment. And both are really important, because when you think about the diagnostic detection piece of it, you have to have a test that is readily accessible, that physicians will actually use because it actually leads to a treatment outcome, and that patients feel comfortable doing because there's an understanding of exactly what the disease risk is and what the treatment interventions are. So I would say that that's almost half the battle. Where we're fortunate is that in a lot of the things, as you guys know, in the diagnostic world, that amount of development has really been, huge over the last 10, 20 years. We've gotten, high throughput sequencing based diagnostics that are now standard of care, certainly in oncology. You have circulating tumor DNA and other aspects, methylation assays that were done by several companies, including Grail. So that has allowed us, I think, to already move there. And then I think what's really kind of the next natural evolution is the diagnostic pieces are already there, getting close. How do we move the therapeutics up accordingly? And that requires both an understanding of the early detection leads to something that's actionable, and the therapies we develop fit that type of patient profile. So that's the large vision, I think, is really early treatment in multiple diseases. And again, that's been something, if I take the oncology example, adjuvant or neoadjuvant therapy, so giving chemotherapy before surgery or after surgery upon new diagnosis, has been one of the things that's revolutionized early therapy in oncology, specifically breast cancer, now lung cancer. So you can imagine that example, but transforming that or parallelizing that to other diseases, autoimmune, cancer, liver disease, even CNS disease. And that's the type of thing where we won't even need to get to the point where the disease is mutated so much that we can't treat it, we're essentially intercepting and preventing that. So that's where I see kind of the broad vision, not only for what somatic mosaicism and CHIP can offer, but I think broadly multiple approaches. And quite excited about that, because that means that then we can get to a point where we can think about cure or ultimately, definitive disease prevention in that regard. And that's, I think, where we're moving in the field of medicine.
Yeah, that's a really exciting future. And I liked at the very end how you specified like more than just curing, like definitive prevention, which everyone, is really interested about the concept of curing. Of course, medicine is a stepping stone progress, and it's been focused on kind of retrospective treatment, which can cover maybe 60% of the challenges. Cures, of course, are the natural next step, but I think that vision, as you had mentioned, detect, intercept, and treat is a really cool kind of paradigm that, I hold the same vision. I think that that's where things maybe should go towards, and I think ultimately it will, on the basis of kind of clinical impact. So let's pivot a little bit into kind of the longevity space. I think kind of extending this now to diseases which, in addition to kind of addressing more of the lethal conditions, focusing on longevity and extension of the health span. So could you share perhaps a brief overview of your opinion on the future of that space, and what are some of like the key challenges and inflection points that you think may be coming our way?
Yeah, sure. Happy to. And I think can kind of separate kind of what TenSixteen is doing versus kind of personal opinions. So I kind of have a background as a stem cell biologist and worked on, hematopoietic stem cells, but also induced pluripotent stem cells during my postdoc. So have some kind of familiarity into the space. It's incredibly interesting. I think the idea that we can reverse certain changes associated with aging or lifespan, and certainly you have concepts like, again, iPSCs or induced pluripotent stem cells that at its core represents that. And I think it provides really nice scientific proof of concept. And I think obviously there are companies now taking those ideas and moving them translationally into how do we get them into patient-based therapies. I think it's really bold, and there's obviously huge applications there. There are some challenges too, I think, in thinking about how in doing those processes do you avoid other off-target effects? How do you create specificity in terms of what you're exactly targeting and so forth? But the field has definitely evolved over the last 10 to 20 years ago, where even this discussion really wouldn't be possible. So I would say, like, from a rejuvenation and kind of cellular senescence reversals, those types of things, it's not exactly what TenSixteen we're working on. We're really looking at kind of a specific biology that relates to aging diseases. But it's another way of tackling, I think, diseases associated with aging. And certainly, I think if you're successful at reversing some of those changes, I mean, the applications are so obvious: Alzheimer's disease, neurodegenerative disease, other aspects. If we can do that and kind of create a younger state of cells, there's obviously applications. So I'm excited to see where it goes. I think, again, there's a lot of things to think about when you think about bringing from the science into clinical proof of concepts, but you have a lot of smart people working on it. So to me, it definitely will be following, the field as it goes forward. Probably not a core area of TenSixteen kind of for what we're going to focus on, but certainly an exciting area nonetheless.
Before we come to a close, Mark, a few rapid fire questions to cap things off. Briefly, can you explain your vision of how big data and omics will be used in medicine in 2030?
Yeah, I think the short piece is everywhere. It's going to be essentially where we take so many different types of data types, and it'll become standard in terms of how we integrate. To me, we're capitalizing maybe on 25-30% of total data, where it was really 5% maybe 10 to 15 years ago. That number is going to go really largely up to the high percentage rates. Again, to me, it's going to be about, how do we make sure we integrate all the different data types and use it in a way that can be digestible, that can be manipulated and analyzed. So to me, it's really the scale of how we use data is going to change.
No, we'd love to hear it. You've given us some great advice for people coming from a medical background, especially like yourself, who were training as physician scientists, and who giving them advice to keep their options open as they explore while keeping that focus. Do you have broader advice for those who are interested in entrepreneurship?
Yeah, I think on the entrepreneurship side, what I would say is, I mean, this is a general piece of advice, but like, do what you love. To me, I think in a lot of times when I talk to certain folks, there's always a sense like, should I do this? Should this look good on my resume? Should I get a PhD because I ultimately want to do this? And I always tell folks that's not the right question, at least in the sense you should ask, do I want to do this because I love it? And I think doing that, you'll naturally find that you'll end up in an area, in a space that you'll have a lot of fun in. I think on the entrepreneurial side, it is really tough. It's challenging. It's not easy being an entrepreneur. And I would say that one of the qualities that I've seen for great entrepreneurs is really a commitment to vision, to really execution, knowing that you're going to be faced with adversity, but your singular focus really is on, I think I can make, or we can make this work. So I think that determination is super helpful. And then again, as I mentioned before, surrounding yourself with people that have done it before, that have made mistakes that you can learn from, mentors that can speak to kind of pattern recognition. So to me, it's the entrepreneur is only as good as the team that they have. And that to me is, I think, a really important concept.
And last but not least, do you have any other closing thoughts or shameless plugs you'd like to share with the audience today?
Yeah, no, I appreciate the idea about shameless plugs. I mean, I would say kind of as a whole, we're in a really exciting time of medicine. And I think it's because, you know, our capabilities for developing drugs has changed. Our capabilities for ingesting data has changed. There's globalization of things that we can do. So to me, I think it's just really an exciting time to be in biotech. Obviously, within TenSixteen, we're really excited about what we're doing, and we are, as a small company, always hiring and looking for great people. So that'll be my shameless plug here, is that we're always looking for folks that are aligned with the vision that we're looking for. So feel free to connect with us. But it's really beyond, it's broader than this. I'm excited about, as you mentioned in closing, where is medicine going in the next 5, 10, 15 years? There are things that are really transformational that are on the horizon, and things that are, you know, going to be really amazing to see, you know, later on. So I think what you guys are doing and highlighting is great to see, because more people, you know, need to understand and see all the different interesting science that's going on, how they seem separate, but they actually fit together in a very large way.
Thank you, Mark, for taking the time to come and share some of that in your story with 47 and TenSixteen today, and for an absolutely fantastic episode. Like you, we look forward to seeing the future of medicine and hopefully helping to create it together. We're very grateful for your time. Thanks again.
Great. Thanks, Chris. Thanks, Michael. It's a pleasure, and I was happy to chat with you guys.